Good morning to everybody. I hope you enjoyed yesterday's evening and welcome to the third day of the symposium. On this Saturday morning, we will continue the scientific program with the plenary session with the key topic of approaches to stability and bioavailability of the biopharmaceuticals. My name is Rok Dreu and I will be chairing this session, and there will be slight change in the schedule, so we will have questions and answer sessions right after each lecture. I encourage participants to be active, and those that are following the conference online, please put the questions in chat and those will be then conveyed to the speakers and answered afterwards. So it is now my pleasure to announce the first speaker. This is Associated Professor Pegi Ahlin Grabnar, who will give a lecture on excipients in biopharmaceuticals from the perspective of formulation, stability, and freeze-drying efficiency. Professor Pegi Ahlin Grabnar is university teacher coming from the Faculty of Pharmacy, University of Ljubljana. Her research is devoted to nanotechnology and lyophilization of biopharmaceuticals. The main focus of her research is on optimizing lyophilization cycles, that is on the shortest possible and just cost-effective processes for drying solutions of biological molecules. We will now follow the pre-recorded lecture of Professor Pegi Ahlin Grabnar, as she is not present here in Gdańsk. Please. Thank you.
Good morning. First of all, I would like to thank the organizers for giving me the opportunity to present this lecture. I will talk about excipients in biopharmaceuticals from the perspective of formulation stability and freeze-drying process efficiency. The fastest growing segment of the pharmaceutical market, and they constitute a multi-billion-dollar industry throughout the world. Protein-based drugs are widely used to treat several diseases, such as chronic viral hepatitis, rheumatoid arthritis, psoriasis, Crohn's disease, and also different types of cancers. The development of such drugs is very challenging and requires great effort and expertise. The preferred dosage form for injectable protein drugs are liquid formulations, but in case that protein molecules are not stable in aqueous media, lyophilization or freeze-drying is the method of choice. With this process, we can enhance stability and extend the shelf life of biopharmaceutical drugs. In the dried state, chemical and physical degradation rates of proteins are significantly reduced. Over the last decade, about 50% of approved biopharmaceutical products have been produced in lyophilized forms, and with the rise in new and highly complex molecule formats, the number of freeze-dried products is expected to grow. Lyophilization cycle consists of three main steps. First step is freezing, where the solution is transformed into ice and freeze-concentrated solution is formed. Second step is primary drying, where the frozen solvent is removed by sublimation. The third step is secondary drying, where the unfrozen water is removed by dissolution. Lyophilization process must deliver a product with acceptable quality attributes, such as low residual water content, short reconstitution time, retention of potency, and also acceptable, that means elegant cake structure. There are two main changes in lyophilization. Success during lyophilization presents the first challenge. Although freeze-drying is a gentle drying method, the proteins are exposed to stresses, such as, for example, cryoconcentration during the freezing step, exposure to ice-liquid interface, and also the removal of hydrogen bonds with water molecules. Therefore, excipients are needed because they protect the protein during freezing, during drying, and also throughout storage in the dried state. The second challenge is duration of lyophilization process. Lyophilization is time and energy consuming process. The most time and energy consuming step is primary drying, which contributes significantly to the total costs of freeze drying. Therefore, lyophilization cycles need to be optimized. Also, in this challenge, the excipients have very important role. A key factor in design and optimization of freeze-drying cycles is the product temperature during the primary drying. Product temperature has a direct impact on the freeze-drying time. Every 1 degree increase in product temperature during primary drying time may result in approximately 13 degrees reduction in primary drying time. Therefore, operating at the highest allowable product temperature is highly desired. The basic theory of conventional drying states that product temperature during primary drying should be kept below critical formulation temperature to avoid macro collapse and to maintain the stability of the protein. This critical formulation temperatures are glass transition temperature of maximally freeze concentrated solution, Tg prime, and collapse temperature Tc, which is typically 1 to 3 degrees above Tg prime of an amorphous formulation. It was recently shown that drying above Tg prime or even above collapse temperature, so-called aggressive drying or fast drying, is not necessarily detrimental to the stability of biopharmaceuticals. Such approach provides an attractive option for optimization of the lyophilization. Formulation development, that means selection of excipients, is very important in case of biopharmaceuticals. The aim of the use of excipients is to provide a stable product with an acceptable lyophilized cake structure and critical quality attributes such as reconstitution time and residual water content. Excipients not only affect the stability of proteins, but they also determine the appropriate operating parameters for the lyophilization process. So, therefore, they are crucial to the development of cost-effective freeze-drying process that enable production of lyophilizates with desirable appearance. Main groups of excipients are stabilizers, bulking agents, buffers, surfactants, tonicity agents. Buffers are added to prevent the pH shift. Surfactants are added to prevent aggregation, to increase protein solubility, and they have also an important role as wetting agent during rehydration of lyophilized cakes. The emphasis of this lecture will be on stabilizers and bulking agents, which have probably the most important impact on protein stability and cake structure. Stabilizers are excipients which protect biological molecules against chemical and physical instability. They can prevent changes during freezing, during drying, and also during storage. During freezing, proteins are stabilized according to the so-called preferential exclusion theory. According to this theory, sugar molecules are preferentially excluded from the protein surface. Stabilization in the dried state is mainly the consequence of two phenomena: water replacement and vitrification. Water replacement describes the thermodynamic stabilization, suggesting that stabilizers form hydrogen bonds with the protein instead of water molecules. Vitrification describes the kinetic stabilization by immobilizing the protein in a glassy matrix and in this way preventing interactions between the protein molecules. In the solid state, an amorphous excipients and protein form a glassy matrix with a characteristic glass temperature. Below the temperature, the degradation of the protein is significantly reduced. Therefore, glass temperature of formulation should be at least 10 to 20 degrees above storage temperature in order to ensure protein stability. Sucrose and trehalose are commonly used stabilizers. They are highly efficient stabilizers during the lyophilization process and the following storage. However, they have constraints due to their low Tg prime, minus 32 for sucrose and minus 29 for trehalose. Therefore, low product temperatures are needed during primary drying, and this results in long cycle duration. Bulking agents have a role in providing an elegant cake appearance. They also increase product mass, act as tonicity agent, and facilitate the rehydration of lyophilized cakes. They should crystallize during freeze drying. If they remain amorphous, they may pose risks to protein stability due to potential crystallization during storage. A crystalline matrix provides a mechanical support for amorphous phase and prevents the cake collapse even if the formulation is being dried above its Tg prime or collapse temperature, so despite performing aggressive drying. Collapse is defined as the loss of pore structure. It is induced by viscous flow of the glassy matrix at temperature above critical temperatures. Collapse would lead to increased reconstitution times and residual moisture in the freeze-dried products, and it is mostly considered as an unaccepted defect. So, bulking agents provide a mechanical support for amorphous phase and prevent cake collapse. Malt products mostly contain mannitol or glycine as bulking agents, since they have high eutectic melting temperatures, mannitol at minus 1.5 degrees and glycine at minus 4. These temperatures are significantly higher than Tg prime of amorphous phase, and thus higher product temperature can be used during the primary drying. Now I will present two of our studies in which we evaluate the formulations with different excipients. First, there is the study with sucrose and sucrose-glycine formulations. The aim of the study was to optimize freeze-drying cycle of two different formulations with monoclonal antibody. The lead formulation contained amorphous sucrose as the stabilizer, and in addition, an alternative formulation was investigated where glycine was included as a bulking agent at the sucrose-glycine ratio of 2:1. In our study, we want to find optimal process parameters, so optimal shelf temperature, which enable the fastest primary drying without impacting product quality. We evaluated critical quality attributes which are specific to lyophilized product: residual moisture, reconstitution time, and cake appearance. An understanding of the thermal characteristics of formulation is critical for the development of an efficient freeze-drying cycle. Therefore, differential scanning calorimetry measurements were performed to determine Tg prime, and freeze-drying microscopy was used to estimate the collapse temperature. With DSC, we determined Tg prime for the sucrose formulation of minus 28.5 and for the sucrose glycine formulation of minus 37.8 degrees. Freeze-drying microscopy is a technique that imitates the freeze-drying process. When collapse temperature is exceeded, there are visible structural changes and macrocollapse can be observed. And here we can see freeze-drying microscopy images of the sucrose glycine formulation. We can see that during drying, the sublimation front, so this border between frozen and freeze-dried material, moves towards the frozen layer, while the freeze-dried layer remains behind it. We can see it as a dark band, and near collapse temperature, some morphological changes occurred within this sublimation front, and we can see large cracks which indicate collapse. For sucrose formulation, the full collapse temperature was at minus 23 degrees, and for the sucrose glycine formulation at minus 28.9 degrees. This is lower temperature than for the formulation with only sucrose, and this is probably a consequence of the amorphous part of the glycine that can contribute to the reduction of collapse temperature. But crystallized glycine provides mechanical support to the amorphous phase, as the crystal lattice of the glycine was still visible down to minus 19 degrees, which was well above full collapse temperature. Different lyophilization cycles were performed. Both formulations were tried in conventional cycle, in which we used shelf temperature minus 20, and in 3 different aggressive cycles, which differ in shelf temperature, so 15, 20, and 30 degrees. In aggressive cycles, product temperature was around minus 25 degrees, which is higher than Tg prime for sucrose formulation. So we determined minus 28.5 degree, and was also higher than Tg prime for sucrose glycine formulation. So we determined minus 37 degrees. In case of formulation with sucrose and glycine, product temperature is also higher than full collapse temperature. In case of sucrose formulation, product temperature is lower than full collapse temperature, but higher than onset collapse temperature. Aggressive cycles resulted in faster sublimation rates and consequently in a reduction in the primary drying times. In conventional cycle, the primary drying time was 48 hours. In aggressive cycle, 25 to 26 hours. So, in aggressive cycles, primary drying has been shortened by almost 50% for both formulations. Here are the results of quality attributes of the products. Cake appearance was acceptable for all formulations. Figure shows cakes of blank, sucrose, and sucrose glycine formulations, which were obtained by conventional on the left and aggressive cycle on the right. And we can see that there was only a minor cake shrinkage after freeze drying. So, although the product temperature of the sucrose glycine formulations was substantially above full collapse temperature, in aggressive drying, collapse was not observed. This means that a crystalline matrix of glycine provides mechanical support for the amorphous phase when it undergoes viscous flow. Surprisingly, there was no collapse in the sucrose formulation either, although the product temperature was near full collapse temperature. Probably the concentration of protein and sucrose was high enough to prevent collapse. Reconstitution time depends on various factors such as morphology, surface area of the cake, and the presence of collapse. It was under 1 minute in all samples, so there was no difference in reconstitution times between the samples prepared with conventional and aggressive cycles. Residual moisture content was in all samples in the desired range, so around 1%. Then we tested physical stability of formulations, and therefore different analytical methods for detection and characterization of aggregates were used. In all cases, we compare pre-lyophilized solution and the reconstituted freeze-dry solids immediately after freeze drying, after 1 month at 40 degrees, and after 12 months at 5 degrees. Spice exclusion chromatography was used to compare the levels of the monomer and the higher and lower molecular weight species. Results in the table show that all of the tested formulations were 99.6% pure or above. That means that the aggressive primary drying did not induce aggregation and was also not detrimental to long-term physical stability. Second method, dynamic light scattering, is a qualitative technique that allows detection of very small amounts of aggregates. The results show that the mean diameters were comparable in all formulations. The mean diameter was around 9 to 10 nanometers. Third method, microflow imaging, was used to detect subvisible particles in the micrometer size range, and we found out that neither aggressive primary drying nor formulation composition had any significant impact on the subvisible particle counts, and were also not detrimental to the long-term physical stability. Here are the main findings of our first study. We have confirmed that by careful selection of shelf temperature, the product temperature can be raised above critical temperatures without compromising cake appearance and protein stability. In case of sucrose formulation, we hypothesized that the concentration of protein at sucrose was probably high enough to increase the viscosity and restrict freedom of movement, and in this way effectively prevent collapse. In case of sucrose glycine formulation, additionally, a crystalline matrix of glycine provides mechanical support for the amorphous phase and prevent collapse. In our study, the primary drying has been shortened by 50%. This presents important time and energy saving, and therefore also financial saving. Here is our second study in which we investigated formulation with mannitol. Mannitol, besides glycine, is the most widely used bulking agent in lyophilized biologics. In freeze-dried formulations, mannitol can form different anhydrous polymorphs as well as mannitol hemihydrate. Mannitol hemihydrate has been seen as potentially negative with regard to the stability of pharmaceutical formulations, since dehydration water could be released during storage. The aim of our study was to evaluate the influence of formulation variables on mannitol polymorphism. We tested 3 different mannitol to sucrose weight ratios, 5:1, 2:1, and 1:1, and sampled with no mannitol. And we tested also 3 different monoclonal antibody concentration, 10, 30, 60, and also placebo. Formulation were freeze-dried, and physical form and morphology of the lyophilizates were characterized with DSC, X-ray, powder X-ray diffraction, scanning electron microscopy, and Brunauer Emmett-Teller method. We performed also the short stress stability study for 1 and 3 months at 40 degrees to assess the influence of different parameters on the model map stability. First, we studied the impact of mannitol to sucrose ratio on formation of mannitol hemihydrate. For characterization of lyophilizates, we used differential scanning calorimetry and X-ray powder diffraction. Figure on the left shows first heating thermograms, which present intensity of mannitol hemihydrate dehydration peaks and the crystallization of amorphous mannitol peaks as a function of mannitol to sucrose ratio. For comparison, there is a thermogram of placebo lyophilizate where these peaks are the most evident. Our results show that the intensity of mannitol hemihydrate dehydration peak decreases with increasing amount of mannitol. In formulation with mannitol to sucrose ratio 5:1, the mannitol hemihydrate dehydration peak was no longer detected. X-ray powder diffraction results are in agreement with DSC results. Here below we can see the example of characteristic X-ray powder diffraction pattern of mannitol polymorphs mixture, and the most important part of this pattern is peak at 18 degrees, which is characteristic for mannitol hemihydrate. Our results show that the intensity of peak at 18 degrees increases with decreasing amount of mannitol. So we can see that there is no characteristic peak in formulation with mannitol to sucrose 5:1. So we can conclude that in case of higher mannitol to sucrose ratio, that means in case of higher amount of mannitol in formulation, less mannitol hemihydrate is formed. Here are the results of the impact of mannitol to sucrose ratio on crystallization propensity of anhydrous mannitol. First, DSC results. Here is the second heating thermogram of lyophilizates. We can see anhydrous mannitol melting peaks, delta, alpha, and beta. The formulation with the highest amount of mannitol exhibited delta mannitol melting peak at approximately 150 degrees, but formulations with mannitol to sucrose ratio 4:2 and 3:3 exhibited this peak at lower temperatures. Figures below shows that propensity of anhydrous mannitol crystallization is the lowest in the sample with mannitol to sucrose ratio 3:3 and much higher in sample with ratio 5:1. X-ray powder diffraction results are in accordance with DSC results. So here is the characteristic diffraction peaks for delta polymorphs at 9.7 and 24.6 degrees, and results on figures show that with increased mannitol content in formulation, the crystallization propensity of anhydrous mannitol is higher. We can conclude that the ratio mannitol to sucrose has an important effect on formation of mannitol hemihydrate and also on crystallization propensity of anhydrous mannitol. Samples with lower mannitol content, so with higher sucrose content, show increased formation of mannitol hemihydrate and decreased anhydrous mannitol crystallization. Mannitol sucrose ratio affects also cake morphology and specific surface area. In the table, there are results of specific surface area in each ratio. The specific surface area of the lyophilized cakes increases with increasing mannitol amount, so from mannitol to sucrose ratio 0:6 to 5:1. Some images show cake morphology as a function of mannitol to sucrose ratio. We can see that in the absence of mannitol, the structure is amorphous. We can see the smooth plates which are connected with large interchannels. The addition of mannitol affects the density of cake structure. With increasing mannitol concentration, the density of the cake is higher. We can see smaller channels and also some micro holes in the plate cake structure. All these changes yield larger lyophilized cake specific surface area as concentration of mannitol is increased. We investigated also the effects of monoclonal antibody concentration on mannitol crystallization. The literature data were inconsistent. We compare formulations with 3 different concentrations of monoclonal antibodies, 10, 30, and 60 milligrams per milliliter, and placebo formulation. X-ray powder diffraction results show that the presence of monoclonal antibodies inhibits mannitol hemihydrate crystallization. As we can see on the diffractogram, the intensity of peak at 18 degrees, which is characteristic for mannitol hemihydrate, decreases with increasing MAP concentration. The effect of MAP concentration on anhydrous mannitol polymorphs crystallization is also concentration dependent. From X-ray powder diffraction and DSC results, we can conclude that the formulation with the highest MAP concentration, so 60 milligrams per milliliter, has the lowest crystalline amount among the tested samples. So we can conclude that lyophilizates with increasing MAP concentration show decreased anhydrous mannitol crystallization and also decreased formation of mannitol hemihydrate. We evaluated physical and chemical stability of the MAP under stress temperature conditions, so 40 degrees. Cation exchange chromatography was used to minimally assess the chemical stability of investigated MAP. Figure shows the difference in main charged variance levels between initial time point and 1 month in black, and between initial time point and 3 months in gray. This difference increases with increasing mannitol to sucrose ratio and increases also with MAP concentration. Although the formulation with a mannitol to sucrose ratio of 3:3 has the highest content of mannitol hemihydrate, it has the highest chemical stability. Physical stability of studied formulations was characterized with size exclusion chromatography. With this technique, we determined the level of soluble aggregates in the submicron size range. Figures show the percentages of aggregation products after freeze drying, after 1 and after 3 months of storage at 40 degrees. And we can see that level of aggregates increases with increasing mannitol to sucrose ratio and also with increasing MAP concentration. The least stable formulation is the one with the mannitol to sucrose ratio of 5:1, although it has the lowest mannitol hemihydrate content. So, in contrast to common opinion, we can conclude that mannitol hemihydrate has no impact on the stability of the MAP used. Increased MAP concentration and also increased mannitol to sucrose ratio in our case mean less molecules of sucrose per molecule of protein. And our additional studies show that the main factor affecting the stability is the ratio of sucrose to MAP, where a lower ratio results in lower stability. Results of our study show that mannitol crystallization is affected by mannitol to sucrose ratio and also by MAP concentration. Increased amorphous part of formulation on behalf of sucrose and MAP promote the crystallization of mannitol hemihydrate. Contrary to the widespread assumption, mannitol hemihydrate content in final product have no impact on the stability of the MAP used. The most important parameter influencing the MAP stability is the ratio of protein to sucrose. In the third part of lecture, I will talk about recent trends in excipients in biopharmaceuticals, so about alternative stabilizers and bulking agents. As we already said, the decisive factor in providing formulation stability is the appropriate ratio between the stabilizer and the protein. Since sucrose has low Tg prime value, high amounts of sucrose in formulation decrease the Tg prime of formulation. Consequently, such formulations require low primary drying time, low primary temperature, and this makes the lyophilization cycle time-consuming and costly. Therefore, alternatives to sucrose and trehalose are needed, which have higher critical temperatures. These excipients are so-called Tg prime or collapse temperature modifiers. Inclusion of such excipients with high critical temperatures allows primary drying at higher drying temperature, which leads to a shorter drying process without loss of the lyophilizate structure. In table, there are some examples of excipients which have higher Tg prime than sucrose. Hydroxyl propyl beta cyclodextrin and 40 kilodalton dextran show in solution Tg prime value of -12 degrees, which is much higher than Tg prime of sucrose. Therefore, we can dry such formulations under aggressive conditions. But in most cases, Tg prime modifiers should be combined with sucrose in appropriate ratio in order to ensure protein stability. Prime modifiers have also high TG, so high glass temperature, so they can also increase TG values of formulations. A higher glass temperature of the freeze-dried product can potentially allow storage at room temperature, which is significantly advantage as we can avoid cold chains throughout the supply chain. Second trend are alternative bulking agents. Mannitol and glycine are commonly used bulking agents in biopharmaceutical formulations, but they require high bulking agents to stabilizer ratios to ensure their crystallization during the freeze-drying process. The essential glycine to sucrose ratio to induce glycine crystallization is approximately 5:2, and essential mannitol to sucrose ratio to induce mannitol crystallization 4:1. Since the crystalline excipients do not provide stabilization of the protein, low concentrations are favored in exchange for higher stabilizer concentration. Therefore, several amino acids were investigated as potential alternative bulking agents. Leucine, isoleucine, and phenylalanine, which crystallized during freeze-drying, were identified as potential alternative bulking agents. Compared to mannitol and glycine, less amino acid is required to form a crystalline scaffold. Pharmaceutical elegant cake was obtained already at amino acid to sucrose ratio of 1:4. Consequently, more sugar can be used in the formulations to generate a higher stabilizer to protein ratio. Results on picture, results of Munich group, show that the amino acids to sucrose ratio 10:40 for leucine, isoleucine, and phenylalanine resulted in acceptable macroscopic appearance for all formulations independent of protein concentration. So this right side of the picture shows acceptable macroscopic appearance. So to conclude, the main goal in lyophilized biologics development is not only to achieve a stable, elegant product, but also to develop a short and efficient freeze-drying cycle. Excipients have an important impact on both, but there is no universal strategy for the selection of excipients, so extensive screening is generally required to identify optimum solution. Finally, I would like to acknowledge the contribution of my colleagues from the Faculty of Pharmacy, Department of Pharmaceutical Technology in Ljubljana, especially to my PhD student, Maja Belušević, and to the co-authors of the publications. I also thank the researchers from LIC Sandu for formulation and analytical support, and to the Slovenian Research Agency for funding. Thank you for your attention.
Thank you. So we have established a connection. Thank you. So are there any questions from the audience, please? Please, Professor Nitowska.
Yes, thank you very much, Professor Grabnar. Really exciting story you presented. And maybe the answer for my question was already on your conclusions, because the last conclusion you presented somehow maybe the answer to my question. When we add surfactants, yes, everything may be changed probably. Of course, it's I'm not talking about the reconstitution step, but at this primary drying, what do you know already how adding surfactants will change everything, or maybe your guess is that still we have the similar relationship that you presented? Yes, thank you.
Thank you for your question. I mean, surfactants should be added in the formulation. The most used surfactants are probably polysorbates, and in some formulations also poloxamers. What kind of, I didn't get the question. I mean, they could have an effect, of course, on...
Yes. So with your experience, what do you think, how much your results may be changed if you add also surfactants during your experiments? Yes, because there are often these proteins, they need also surfactants in the formulation.
Yes, we have also surfactant in all formulation. I mean, polysorbate.
You have in your so in your formulations that you presented, there were always surfactants.
Yes.
Oh, okay. So this I didn't catch. Thank you very much.
Thank you.
So, do we have any other questions? Maybe from chat. Okay. So my question would be. So you have here presented how the ratio between the bulking agent and the stabilizer actually influences the efficiency of the crystallization process, which is important for the cake morphology. So my question here would be, you found out that you have to have a rather high ratio of bulking agent to stabilizer, and that reduces the amount of biological molecule that can be introduced into the dosage form. My question here is there any other technological approach to induce crystallization of the bulking agent during the lyophilization process?
Yes, because there are also, of course, the process parameter of the process. And the condition of freezing step is very important, and addition of annealing step is that process which increase the probability of crystallization of mannitol or other bulking agents.
Thank you. And in one of your study, you have also mentioned the microflow imaging technique. So I'm not so familiar with this technique. I know you have used it for the subvisible particle count, which is of course important for such dosage forms, for parenteral dosage forms. And as we know, in compendial methods are light obscuration test and microscopic test. So is this microflow imaging technique used only for the development, or do you see is the reliability of such technique high enough, and in the future we might expect that it will be used also as a compendial technique within the USP and European Pharmacopoeia?
I think that it is very useful technique. I mean, this is the particle analysis technique that uses flow microscopy to quantify particles in a solution based on size. And this technique is already used in the biopharmaceutical industry to characterize the subvisible particles from approximately 1 micrometer size to 50 micrometer size. And I think that, I mean, it could be instead of light obscuration.
Do you know that whether the size estimation of the particle within this the sample area, measuring area, is based on image analysis technique or some other approach is used?
Yes, it is also based on image based technique. I mean, we can distinguish particles of the protein and maybe the droplets of the silicone oil and some aggregates, so we can distinguish such things in the sample.
I'm asking this because the major limitation of light obscuration technique, which is compendial technique, is that it does not know the shape of the particle. It just assumes it is round, and then we have a large errors based on shape of the particles. Thank you. So maybe some other questions. We have still 2 minutes time for discussion. If no, we can now continue with our program. Thank you again. So our next speaker in this morning plenary session is Professor Dr. Andreas Zimmer. I guess we all know Professor Andreas Zimmer very well, as he's a frequent active participant and an organizer of a successful CEST conference in 2010, which was held in Graz. So nevertheless, let me shortly introduce Professor Andreas Zimmer in a few words. So Professor Zimmer is coming from Karl Francis University, Graz, where he is leading the Department of Pharmaceutical Technology and Biopharmacy. The research interest of the department is focused on pharmaceutical nanotechnology and covers all kinds of colloidal drug delivery systems and dosage forms. Professor is a winner of Young Pharmaceutical Investigator Award from OIFEPS and of prestigious Phoenix Award. Today he will give a lecture on a very contemporary topic and will reveal how delivery systems enable fight against COVID-19 and how the technology which is used is not so new as people might think. So Professor Zimmer, the floor is yours.
Yeah. Okay, thank you, Rock, for the very nice introduction. I liked it very much. I also would like to thank our president and all the organizers for inviting me here to come to the conference. And as this conference was starting in organization and I was asked to come here to Gdańsk, I found it a very nice idea, and I thought about, well, this would be only possible if we have a good vaccination strategy. And so I had the idea to present today some new details about the messenger RNA vaccines. And as you already mentioned, many people are asking, how is it possible that we can develop and manufacture that vaccines during a short time? Basically, it was done within 1 year. So I will actually try to convince you that it's not a miracle, so we will see how it works. This presenter is not. This is the first thing which is not working. No. May I have to switch it on? Okay. Thanks. I think, as everybody knows, we have here a nice cartoon from the new coronavirus. WHO declared an outbreak on this virus end of January last year, 2020, and a couple of weeks later, WHO claimed the pandemic situation. And still, as you can see here in the audience, we are still in this pandemic situation since that time. The main structure of this virus, as you can see it here, is a so-called positive sense single-stranded RNA virus. And this was the main reason why it makes it also quite easier and straightforward to use messenger RNA as a new vaccination strategy. As you can see here, the total sequence, RNA sequence from this virus consists of about 30,000 nucleotides. And here on the right side, as you can maybe see it here, it's the spike protein, the sequence of the spike protein, which is used for the vaccines. It's approximately 3,800 nucleotides large, and you can see it here from the picture on the left side. This is a more or less naked spike protein. On the right side, we have the glycosylated version, so it's a fully glycosylated protein. Just a few words about the messenger RNA technology. Indeed, it's not a new technology. As you can see it here from the graph, we already started in the year 1961 with the discovery of the messenger RNA. And the basic concept of messenger RNA to have been used as a vaccine was already published in 1990, so many years ago. And the story continued approximately 10 years ago when companies started to develop these vaccines, especially for influenza, flu, and other viruses. So this started in 2012, and so we had approximately more or less 10, 12 years experience in terms of preclinical and clinical development phase for such kind of vaccines. And as you can see it here, the latest circle, 2020, the companies, BioNTech and Moderna, immediately stepped into the strategy to develop the messenger RNA vaccine after the sequence was published, and this was very early, also in January last year. Both companies are using a so-called modified RNA, uracil modified RNA, to achieve a higher stability of the messenger RNA. And this is also not a totally new strategy because it is known for a long time and was published also in 2008 that this could be a good opportunity to stabilize the messenger RNA. And RNA indeed is not such a stable molecule, though it needs strategies to improve the stability. And more or less 1 year later, on 2nd December 2020, the UK authorities approved the Pfizer-BioNTech vaccine. 1 week later, FDA did the same with an emergency use. And you may have heard that very recently in August this year, FDA issued also the regular usage of such vaccine, and currently Moderna is applying to do the same. Here we have both vaccines which are on the market currently. So the Pfizer-BioNTech Comirnaty vaccine is based on a modified RNA sequence, which approximately 4,200 nucleotides. Pfizer-BioNTech is using a so-called 2P design, which means that they have introduced some mutations with including 2 prolines. And Moderna is exactly doing the same. The Moderna vaccine Spikevax has actually the same technology, and this 2P design was already published before to improve the stability of the peptide sequence. So Moderna Spikevax is a little bit smaller. It includes 4,100 nucleotides. They are encoding for the full-length SARS-CoV-2 spike glycoproteins, having actually both mutations as well. So in principle, these messenger RNA-based vaccines are approved now in more than 96 countries worldwide. In principle, we have 3 vaccines because Moderna gave the license to Takeda in Japan to do clinical trials in Japan. So this was not the first approach from Moderna to do it in Japan. So Takeda stepped into this business, but other companies are following this strategy. And so we will have the next messenger RNA-based vaccines coming from Valvax, which is a Chinese company. They already finishing phase 3 of the clinical trial, so we expect the registration and approval of that vaccine pretty soon, until the end of this year. But obviously, Pfizer-BioNTech and Moderna are working on updates of these vaccines. as far as it is known from the clinical trials, we have seen that Pfizer-BioNTech starting to improve the vaccine in terms of storage stability, and you may have heard that this vaccine has to be stored at low temperature between -80 and -60. Then they got an update to have also stability at slightly warmer temperature. Still, it's in reality temperature for the storage conditions is a challenge, and also Moderna is working in the same direction. All companies are also trying to monitor the situation of the current mutations and variants, and we will see definitely updates from the virus vaccine strategy pretty soon. Other companies like CureVac are in phase 3 and starting also collaborations, CureVac with GSK to have double compound vaccines. Sanofi Pasteur is doing the same. And we have one company, Cepheus Vaccines, which is collaborating with the Ghent University. They are just working on the next generation of the RNA vaccine, so-called self-amplifying RNA, but I will come back to that point a little bit later. Anyway, all these companies are using lipid nanoparticles for the formulation of the messenger RNA, and this is definitely also not a new situation and a new technology. You all know that liposomes have been published and discovered by Bangham in 1964. The lipoplex is basically the same what Pfizer-BioNTech and Moderna are using. The technology has been published in 1987 by Philip Felkner. I just found the publication, you can see it here on the left side, where the first time was lipofaction was published, and definitely later, also 1991, first time Rainer Müller published solid lipid nanoparticles, and later the nanostructure lipid carriers. You can all see here different structures on the slides, and this is definitely not such a totally new story. In 2018, so approximately 3 years ago, the FDA also approved the first drug, which was not a vaccine. So this drug is called Onpattro. It is directed against polyneuropathy, and includes so-called small interfering RNA. So it's not a messenger RNA, but in principle from the chemistry, it's pretty much the same, but the molecular weight is smaller and shorter molecule. Anyway, the company for the first time did something really challenging, especially also for the authorities, for the FDA, because they applied with a drug application to be the first drug in class, and including the first galenics and the first technology. So it was more or less a double nightmare for the authority, but actually they did a good job. And as you can see it here on the right side, it's including lipid nanoparticle where the RNA is encapsulated, and it's using pegylated lipids which are decorating on the surface to get it more hydrophilic, and they include cationic lipids and helper lipids, cholesterol as well. And that's pretty much also the composition what we see with the vaccines technology today. Just a few words about the cationic lipids, which have been used and improved by the FDA. There are lots of substances on the market right now. It's a big business for all the companies, Avanti Polar Lipids and others, which stepped into the synthesis, providing pharmaceutical industry with that kind of excipients. You may know that DOTAP and DOTMA, as you can see it it here on the slide are probably the first compounds, DC-col and others, which have been used since, let's say, more than 20 years in such a business. What happens when you combine an RNA or DNA molecule with a liposome, a pre-formulated lipid particle? It will change the structure as is shown here. I took this presentation and this picture from a nice publication from Brigitte Sternberg, and she was the first author who claimed really that the structure of the lipid particles and liposomes is changed by the interaction between the RNA and the cationic lipid. So we will end up in phases, lamellar phases, and so-called spaghetti-like structures, as you can see it here on the image coming from a freeze fracture electron microscope. Also in our group, in my group, together with Christa and Noe, we try to synthesize and use aminohexyl cholesterol as a cationic lipid, and actually this is more than 20 years ago. This was quite successful. I don't want to go into these details too much, but as you can see it here on the right side from the graph, the upper line with the dark triangle is the protected nucleotide sequence, protected against enzymatic degradation, and the non-protected sequence is here on the bottom line, is degraded pretty fast by nucleases, and that's basically the strategy which is used also for formulation to keep RNA stable and most efficient within these lipid nanoparticles. So coming back to the vaccine formulation, which was presented and claimed from Pfizer-BioNTech, this company is using cationic lipid, as you can see it here on the right side. The nitrogen is a centered molecule which is charged pH dependent. This is a strategy what all companies follow so far. So we have a deep pH dependent positive charge in that lipid. Together, as they use also polyethylene glycol, pegylated, so-called pegylated lipids, as you can see it here. This is the second one. Then all companies are using DSPC as a twitter-ionic amphiphilic lipid, and further cholesterol is used as a main structural lipophilic compound in the lipid nanoparticles. And as we have heard in the first talk, also sucrose is used in that formulation, and sucrose works as a cryoprotectant, as we have seen before here today in the first talk. That's basically what it is. It's not from the composition of the excipients. It's not that complicated. And on the next slide, we see pretty much the same coming from the Moderna vaccines. They are using a little bit slightly different chemical compounds, but in principle, it's more or less the same. Yeah? They also have sucrose as a cryoprotectant, a little bit different buffer substances, but in principle, both vaccine composition are quite similar. For the quantitative composition, also both vaccines are mainly based on the cationic lipids in combination with cholesterol. The cholesterol and cationic lipids together is the main part of the lipophilic nanoparticle, and pegylated lipids, which are decorating on the surface, counting probably 2%, so it's not that much, but this is something what we already know from pegylated liposomes. Here we have the same situation. The total molar charge ratio between the cationic lipid and the messenger RNA, which is included, slightly differs from one formulation to the other. It's also a little bit different with the Onpattro but in principle, they all follow a strategy between having let's say, a charge ratio between 2:1 and 5:1. So this is, I think, basically very common for all these formulations. CureVac is something doing a little bit different. They claimed so-called RNA active technology. These vaccines have been developed for oncology, so for cancer vaccination during the last 10 years. And this company tried to adopt this also to the new coronavirus. They currently joined a joint venture with GSK, and also in this situation, they probably switched to the lipid nanoparticles. But RNA active technology means that they try to get a combination between free RNA and protamine complex RNA. This is a little bit different from what we have seen from the lipophilic nanoparticles so far, but might be also promising in the future. You may know that we are working exactly in the same direction. And yesterday, from my PhD students, we have seen also the potential of that strategy. So we will see the outcome, how it works in the future. Okay, coming back to this drug, which was already approved, it's not a vaccine, but also from a quantitative point of view, from the composition, we have the same main compound, is a cationic lipid and DSPC and cholesterol. So from in principle, these formulations are very similar and have been developed for the, let's say, last 10, 12 years. So it's really not a miracle to expect that this works also for the messenger RNA vaccines so far. So what happens when we go with the lipid nanoparticles in vivo? What is happening and what is the in vivo mechanism? Basically, what happens with all kinds of particles, what you inject into the body, it will definitely interact very fast with proteins coming from plasma, or if you're going for an intravenous injection, the vaccines are injected into the muscle, but could be also there are technologies available to apply the vaccine into intradermally, so under the skin. Actually, they will interact with peptides and proteins which are surrounding the lipid nanoparticles, and this is called opsonization. ApoA was shown as a lipoprotein to trigger a redistribution of the lipids within the lipid nanoparticles, and it's also facilitating the uptake and the endosomal escape. And this endosomal escape, which comes next within the cell, is one of the most crucial steps for the efficacy of the vaccine, because as we know from calculations and from investigations, it is known that approximately only 3 to 5% of the RNA can really escape as an intact molecule and can be processed further into the spike protein, which is later expressed here at the surface and can interact with so-called T helper cells or cytotoxic C cells to improve and to trigger the next step of immune response. A few words about the manufacturing technologies. It's basically all about microfluidics, what we have learned during the last 10 to 20 years. All companies are using a so-called modified ethanol injection technology, which is based on a static mixture, and you are mixing in principle an organic solvent with a water-based solvent, a buffer, And this is also not a new technology. I found this publication which was published in BBA at, I think it was 1973, showing the first time the potential of such kind of ethanol injection. So this is something what is widely known also for liposomal formulation since many years. What happens is pretty simple. All the compounds are dissolved in one of the solvents. In ethanol, could be also in the water, depending what it is. If you have a buffer substance, it goes to the water phase, of course. And during this mixing procedure, the solvent polarity is rising, and it could be also done in a step-to-step manufacturing, but in principle, it's a very simple, straightforward strategy. And finally, after mixing all the compounds, the nanoparticles are finished. This was, as I mentioned, the first time approved by the FDA with that kind of particles which have been used for the Onpattro. And as you can see it here on the right slide, we have this more dense structure from the lipid particles here. As you can see it here from the electron microscopic image. And if you don't have the RNA inside, we will still end up with pegylated liposomes. That's what it is. This microfluidic devices can be even more developed and elevated technology, combining different solvents, different buffers. You can also create a pH shift, which might be quite useful for combining a cationic lipid together with the RNA and then doing the surrounding encapsulation with the other helper lipids. And that's something what works pretty simple, and it's a straightforward technology and can be easily upscaled. What you see here on the left side is a nano assembler, I think a benchtop instrument, something what we have in the lab, not very large, it's static mixing devices. And from that technology, which is probably able to manufacture 1 liter of the vaccine per hour, you can easily scale it up with the same technology. As you can see it here on the right side, I took this picture from Polymoon, which is a company based close to Vienna in Austria, and they did a lot of product development, and also they are in the business as contract manufacturer for doing GMP production. And the production unit is not that large, so it's pretty tight and simple GMP facility. And this facility will allow the manufacturing of about 250 liters in 1.5 hours. Then it's basically finished and can go to fill and finish as a next step as well. This technology in principle is also not that new. It's used for many years also in synthesis and chemistry industry. And we did more or less the same a couple of years ago together with an Austrian company, which is called 1A Engineering. And they developed such kind of microfluidic device based on a capillary reactor, as you can see it here, and we further investigated what happens in the reactor and what is the timing and production time and so on. And as we found out, it's basically all done within a couple of milliseconds. So starting this mixing procedure after 200 milliseconds, you will reach constant size and size distribution. So basically after a couple of seconds, the product is finished. Okay, a few words at the end of my presentation goes to the next generation messenger RNA vaccines. Here we have seen that Pfizer-BioNTech and also Moderna working on an update in terms of getting more stable vaccines. And as we have seen in the first presentation today, lyophilization is really an option. I think it will be possible to be lyophilized, but we need probably more time to develop this lyophilized formulation. As we have heard today in the morning, it needs extensive case-to-case investigation to improve this situation, and lyophilization is not an easiest production technology so far. Anyway, including adjuvants is always a topic for having new vaccines. So far, we know that the lipid nanoparticles are doing not a bad job, so we probably have not this historic options available, or probably it's not necessary to include alum and other things to improve the efficacy of the messenger RNA vaccine so far. But what we know is that Pfizer-BioNTech and also Moderna are using different doses. So Moderna has, for the single injection, is using 100 microgram, and Pfizer-BioNTech is using only 30 micrograms. That was the reason why also the FDA asked Moderna to lower the dose, and having a lower dose is always an option to have to improve the whole formulation anyway. And this is also the direction where self-amplifying RNA vaccines will be going into the next generation of these vaccines. As I mentioned, there's one company, Cyphios Vaccine, together with the Ghent University, which is working on that new technology. A few words about self-amplifying RNA vaccines. I have prepared this slide. As you can see it here in the upper part, the classical messenger RNA will just have the immunogen be translated in situ, and the self-amplifying RNA includes production unit, or let's say replication unit, coming from an alpha virus, and this replication machinery, which is translated, has the main job from this production unit within the cell will be the amplification of the RNA, which is also included in the sequence, and therefore it is probably not necessary to have such large doses. So we can go down to lower doses, maybe 100-fold lower doses are really achievable, and it is also known that we might have less side effects. This is that kind of technology. Okay, some conclusions. I hope I could convince you that messenger RNA vaccines are not that new. You probably have known this, and we are working in this field since more than 30 years, basically in nanotechnology and liposuction, especially in basic research, also in academia. The main formulation technology was approved by the FDA and EMEA before. And my personal view is that this technology, especially also going to DNA technology, will be further expanded very recently. And I think the future is should be quite nice. And we have actually huge options, especially in oncology, to use the same technology, but also in other fields like neurodegeneration and other diseases. So I would like to thank to all of my team members and PhD students who worked in that field during the last 20 years. There is a lot of literature at this topic. As you may know, you can have an update every week. If you are interested in having current literature, please let me know. I can send it to you. And thanks a lot for attending here the session, and also thanks a lot for joining us in a virtual way and online. Thank you for your attention, and happy to take your questions.
Professor Zimmer, thank you for this very educational talk. It's very important that we are educated in a proper way, that we have the latest information from this field. So as we have to educate people, as we know, we are following the paradigm science to the people, but people have to understand science. So there are many cases when oversimplifications are made, and this is one area where we should have to be clear to avoid any confusions, to facilitate the vaccination process. So people have to understand that. So this is why this lecture is very important, and of course it's now open for discussion. Maybe two or three questions could be still. So we have a question from the chat, so from the online participants, please.
Yes, so we have a question from Adelian Szewczyk, and I quote, "Thank you for an excellent presentation. Regarding to ethanol injection method, may I ask you how you confirm the successful encapsulation of RNA inside the nanoparticles, and how efficient is that process?"
Well, in principle, as far as I know, and as it is published, the process is very efficient. Of course, we need some purification steps, which includes dialysis to get rid of the ethanol and the organic solvents. So this has to be, let's say, separated from the lipid nanoparticles, but this can be done with dialysis, pretty simple step. And indeed, since we have this increasing polarity, as I've shown in one of the slides, the complexation between the cationic lipid and the negatively charged RNA is done in a quantitative manner.
So, please, Professor Breiner.
Professor Zimmer, thank you very much for a very interesting and informative talk. As you indicated, there are several companies at the moment working along similar lines as Pfizer, Moderna, etc., etc. Where do you think that a major difference or improvement could be made? Is that on the efficacy side, or on the safety profile, or in the stability profile? Because those companies, of course, think of an incentive that will improve or increase their market share ultimately.
Yeah, you're totally right. I think the first challenge was, as we have seen it early last year, that Pfizer BioNTech needed such low temperature for the storage. So this was the first attempt to improve the vaccine technology to come or to be a little bit warmer. Yeah, -20. Moderna was a little bit better, but still -20 for the supply chain into other countries worldwide is a challenge, still a challenge. So I think this is a first step which has to be improved to get higher storage temperatures. This is the first one. Efficacy in terms of endosomal escape is also a question. If you have only 3 to 5% of the RNA, which is really available for the whole translation process, you can have a huge impact just by improving this technology. Therefore, you need probably further developed cationic lipids or other compounds, additional helper lipids, which facilitates these endosomal escapes. This is also a strategy. And of course, we have DNA technology, which might be an option as well. And as I mentioned, the self-amplifying RNA could be really an option, because when you can lower the dosage, let's say from 100 microgram to 1 microgram, that's a huge step. When you compare this to a classical, let's say, small compound aspirin, yeah, where you have to take every day 100 milligram aspirin, and you could lower down to 1 milligram aspirin every day. This would be a huge improvement. Yeah.
Thank you.
We have another question from Dr. Schliaut.
It is not a question, just a comment. So thank you very much. It's really very informative. By the way, I will touch base on that about the history of mRNA, but not in that excellent details. You are the expert in this field. I am wondering actually why the media in Europe in general, they are not mentioning all these details, because the perception in the public domain is totally different.
It's different, yeah. It's all about education. Yeah. I think so, yeah. So if you talk to representatives from the media, they don't know about it. That's the main issue. If you don't know about these details, you can publish it.
Therefore, we can't repeat this enough.
Yeah.
Thank you very much.
Yeah, you're welcome.
So I guess we have concluded this session. Thank you again, Professor, for this excellent.
I will apologize for those who are online, but probably you already are used that we are coming back from our breaks a little bit later. So the nice coffee and cakes you must regret, but unfortunately we also never have enough time to discuss the topics here. I will start the last session, and we have one short talk and 5 video presentations of the posters. First, I would like to ask to present, this will be a pre-recorded presentation, to present results. Pavlína Komínová from University of Chemistry and Technology in Prague. She will present the effect of polydimethylsiloxane additive on process of filling corn starch fillers into hard gelatin capsules. We will discuss, we'll have questions and answers after also video presentations of the posters. So at the end of this session. May I ask for the presentation?
So at first, I thank you for my introduction and give me the floor. So good morning, ladies and gentlemen. My name is Pavlína Komínová, and as already mentioned, my contribution deals with the topic of hard gelatin capsules, and it is focused on impact of polydimethylsiloxane present on capsule filling of cornstarch fillers. I'm sure that each of us has personal experience with hard gelatin capsules, and it isn't necessary to introduce them in detail. So just a few words about them. Hard gelatin capsules belong among solid dosage forms that are primarily used for oral application. And as their name indicates, the main ingredient they are made up of is gelatin, and they consist of 2 parts, body and the cap. And they are usually filled with various types of particulate systems, such as powders, granules, or pellets, and so on. The whole filling process can be simulated on this simple flow chart with highlighting of the main important step, the filling of the capsule body with the powder or any other particulate systems. For the smooth running capsule production, it is necessary to achieve the effective filling, and this condition is reached with the powders having the rheological properties at a certain level of acceptability. And regarding the fact that regarding the variability of particulate system in this complex parameter, the capsule's body filling becomes also the very critical parameter. And it is even more complicated if due to the fact that we have a number types of filling machines. For example, the simplest one, manual filling machines or semi-automatic filling machines. Based on these examples, it can be concluded that external factors that are connected with the filling machines vary and affect the rheological properties in different manner. And let's go a little bit deeper and talk only about one type of filling machine. As a nice example, I choose the most often used filling machines, the automatic filling machines. These devices works mainly on two principles, dosing disc or dosators. And if we focus on the market parts in the text that are connected with the phases where the rheological properties are more crucial, and compare these phases specific for the devices using dosing disc principle with phases specific for devices based on dosator principle, we find out, or it is obvious that these two operation principles aren't identical, and similar the requirements on the flowability of used powders. This overview in this slide just summarizes the basic differences. In case of dosing disc, the creation of the slug is achieved by repeated application of normal stress. In contrast, just one application of compaction is needed in case of dosator. Furthermore, the strength of the compact is more important in case of dosator due to the necessity of holding the slug inside the dosator during its transfer from the powder bed above the capsule body. And research up to now has also shown that the range of powders that can be filled with the dosing disc is wider. And this is especially valid for powders with good flowability that cannot be used in case of dosator due to the weak or very firm of created slug. In other extreme, very cohesive powders cannot be used as well due to the very complicated ejection of the slug from the dosator. And what does this overview means, or what does it imply? It implies that if pharmaceutical company wants to fill one formulation with different devices, let's say the filling problems can arise as the formulation might not meet requirements that are specific for the devices, and the problems can arise. And in order to outline how this theory can be applied in practice, the second part of my presentation is dedicated to case study that is focused on studying real mixture intended for hard gelatin capsule filling. And a few words about the mixture. This mixture composed of polydimethyl liquid siloxane, liquid polydimethyl siloxane that is known as a dimethicone, cornstarch, and API. The process of its preparation is 3 stages. In one stage, the so-called premix is prepared and includes the dimethicone and defined amount of cornstarch. The second stage includes preparation of active mixture from another amount of cornstarch and API. And in the last step, the left amount of cornstarch is added to active mixture and premixed to obtain the final mixture. And the main attention or the main goal was to define and describe the effect of dimeticon and logical properties of included premix or corresponding systems. It means systems including dimeticon and cornstarch. To achieve it, we studied the effect of dimeticon amount, preparation technology of premixes, and effect of used cornstarch type. Also, we studied the logical behavior of final mixtures, including the studied premixes, where for all of them, the total composition was maintained similar. In general, cornstarches can be got in native or partially progenialized form, and both of them were included in this study. As a representative of native starches, the starch with brand name merizet was chosen. The remaining two, Licatap and Starcap, belongs among partially progenialized one. We opted for this tree because of their different particle characteristic. How we can see in these SEM images, they differ in their particle size, shape, and also particle size distribution. In order to evaluate the effect of mixture composition or amount of dimeticon, several premixes with various dimeticon concentration were prepared, precisely with 0.5 to 6 and 10% of dimeticon. The premix with 6% of dimeticon is used in original mixture composition. In case of preparation technology of premixes, 3 ways were investigated. First one, the original, was simple mixing using 3-dimensional motion, and the mixing condition was set to 80 RPM and 8 minutes as a mixing time. The second approach, it is called sieving approach, includes incorporation of sieving step in course of mixing. And in the last step, compaction, or last approach, compaction approach, the premix was after mixing compacted with the compaction load 75 megapascals, and after that pushed through the sieve. Nowadays, number of parameters and method can be used to describe rheology properties of powders, and for example, the traditional one are repose angle, flow through the orifice, or car compressibility index. In this study, we use the FT4 powder rheolometer that you can see in this picture, and this device offers several methodologies and methods that can describe powder behavior in case of their bulk, dynamic, or shear properties, and study their behavior during pharmaceutical processes such as tableting dye and capsule filling, or ejection, mixing, and many others. So it gives us a more comprehensive description of powder behavior. To avoid surface properties of studied mixtures, shear measurement was applied. Its principle based on the movement of 2 powder layers against each other when one is prevented from a movement. The results are therefore a reflection of shear strength of powder and surface friction acting between particles. This test provides several parameters to define the friction properties, and we choose the effective angle of internal friction when the lower values of this angle means lower interparticle friction. And let's move to the results. In this graph, we can see dependence of effective angle of internal friction on a concentration of dimeticon in premix. We can see that the effect of dimeticon or impact of dimeticon is affected by used cornstarch type. In case of starch mRNA, having smooth particles, the dimeticon is absorbed on the surface and the angle value increases with the dimeticon amount. For starch Licatab and StarCap, local minimum is visible. And due to their surface unevenness, the dimeticon is incorporated into cavities at first, and for the surface, it is available after their filling. It means that until a certain amount, the dimeticon improves surface properties. However, in general, the increasing trend of the angle value with dimeticon concentration is clear. How we can see in this SEM images, for premixes containing starch mRNA and 0.5 and 6% of dimeticon, the liquid dimeticon on the surface causes contact area and work as an agglomeration enhancer. And this phenomenon is more pronounced with its higher concentration and is connected with the higher friction. The second used measurement within the research was compressibility measurement. This measurement was applied or selected to define behavior of mixtures as a compact or coherent powder bed. And during this test, the powder is consolidated by vent piston until the specified normal load, usually 15 kilopascals. And the parameter called compressibility is obtained. This parameter measures the percentage change in volume of powder bed and indicate the self-arrangement of powders. And this parameter can also reflect the flow units, character of your flow units that are responsible for the powder flowability. And in general, lower value of compressibility means better particle self-arrangement and the mixture shows better flow properties. And how we can see in these results, presenting as a dependence of compressibility on applied load. Again, there is an impact of used cornstarch type, and in respect to applied preparation approach, different agglomerates arise. How we can see in this SEM images for premixes of again starch mRNA prepared by all three approaches, the agglomerates evince different character depending on the used preparation approach. And also they have a different flowability, where the inclusion of sieving and compaction approach leads to worse rheological properties. And the last applied measurement, aeration measurement, simulates processes where the air is intentionally introduced into powder bed and defines the energetic demands. But most importantly, it can also define the cohesive forces that exist between particles and are responsible for the agglomeration. Therefore, we use this measurement as a measurement of agglomerate strength via the parameter that is called rated energy with respect to the velocity of the air, and the lower value of rated energy means lower cohesive forces between particles. And if we move to the results again, we can see that with increasing dimeticon amount, the violet curve, the lowest concentration, and the blue one, the highest concentration, the higher energies and stronger agglomerates are obtained. In case of all StarCap premixes, the lowest energies are observed and decreases with the applied air velocity. And this indicates the highest weakness of included agglomerates and their destruction. And if we focus on the curve trend, we find out that for premixes with merizet and licatab starch, the premixes evince similar behavior from the strange point of view, even though the starches have different particle characteristics. And how these all findings affect, or findings about premixes affect the final mixture's behavior. The compressibility measurement shows that the dimethion amount in premix and use cornstarch type have the most significant contribution on the behavior of final mixtures. And it is connected with the agglomerate strength, what is obvious from the aeration measurement, where the agglomerate strength is more pronounced for the mixtures having the premixes with higher dimethion concentration. On the other hand, there is an infinitesimal effect of preparation method, mainly in case of mixtures including premixes, including low dimethion loaded premixes. And this was the last slide with results, bringing me to the conclusion. In this study, the formulation for capsule filling consisting of cornstarch, dimethion, and API was studied, and the main attention was paid to the influence of cornstarch type, dimethion amount, and method of preparation on rheological properties of included premixes. It was found out that dimethion pronounced the agglomeration of cornstarch particles, and the distribution of dimethion and its agglomeration effect is affected by all investigated parameters. As a result, agglomerates with different rheological properties were obtained. And this finding essentially defined the possibility of dimethion application to modify rheological properties of pharmaceutical formulation. And it is of great practical experience, as it can allow transfer the filling process between filling devices without need of change of marketing product authorization. And the results also shown that dimethion can serve as a factor that compensates the variability of cornstarch fillers. And this is all from me, and I thank you for your attention.
Thank you very much. As probably some of you noticed, some miracles also happen in our symposium, because I was informed that you will be online, and suddenly you appeared. So transportation from Prague is really fast. Thank you for coming.
Process everything.
Yes, and of course we will have questions to you at the moment. So please, it's open for discussion. Professor Breitkreuz.
Thank you very much for your interesting talk. I have a question. Do you know about the fate of the silicon particles afterwards in the capsule? Do they interact with the capsule shells? Did you investigate this? Does the shell become more soft afterwards, for example?
No.
Okay.
I don't have a question, but rather a remark. Since you are doing also the measurements with the shear cell,
Yeah.
then one idea would be to use a compaction load at the beginning of the measurement, which reflects the actual load which is used for a specific capsule filling machine, and out of that you can then estimate unconfined yield strength for that material, and that could be a sign of the plug stability in terms of later capsule filling, not just from the rheological point of view, but also from point of stability of the plug, which is especially important, as you mentioned, for that dosator technology. Just a suggestion.
Yeah, thank you very much.
For me, it would be very interesting to compare your results, or at least some of them, with the results if you use Simeticon. So Simeticon contains also silica. So it would be also maybe the other effect, and also in gastrointestinal tract, maybe it behaved in different way. Did you think about that?
Not yet, but it's a nice idea because they have also a different viscosity. So I think that this parameter will be a big effect on the behavior of the surface properties.
And you combine both the effect of silica and dimeticon. No questions? Yes. Thank you very much once again for coming also. So we will start now our video posters presentation. The first presentation comes from the group of Professor Jachowicz from Kraków, and Adam Pacławski will present data-driven modeling of dissolution process of poorly soluble drugs from powder systems.
Hello everyone. In January of 2020, first molecule designed by artificial intelligence was introduced into first phase of clinical trials. By making short summary, it was pointed that the use of data-driven models allowed to reduce time and cost of research 5 times. My name is Adam Pacławski, and I would like to present very shortly how data-driven models could be applied in pharmaceutical technology field. The general goal of research was to develop and analyze predictive model for dissolution process of poorly soluble drugs from powder systems. The database applied to develop predictive models was created based on in-house data, expanded by information harvested from literature. In total, database had over 2,600 data records and was encoding over 300 binary or ternary powder systems. Number of input variables was 3,153, and there was just one output variable, which was amount of API dissolved after a specified time. In the next stage, feature selection and model development process was performed. Models were tested according to 10-fold cross-validation method, and from the all applied tools, the best was TPOT model, which is genetic programming method. The RMSE was below 16 and R² was equal to 0.75. In the end, model was analyzed using Shapley additive explanations method, and the results are presented in figure 1. From the most important variables, we have time, preparation method, descriptor for the excipient, amount of the excipient in formulation, and descriptor for API. Thank you for your attention, and if you have any questions, I will be happy to answer them. Here is also my email address if you need more information.
The next presentation also comes from the same group in Jagiellonian University in Kraków. Ewelina Łyszczarz will present the topic optimization of electrospan or dispersible films with aripiprazole.
Hello everyone, my name is Ewelina Łyszczarz and I work in the Department of Pharmaceutical Technology and Biopharmaceutics at Jagiellonian University Medical College in Kraków. The aim of our study was to optimize the composition of the electrospinning solution, the process parameters, on the fiber's morphology and their dispersible films with aripiprazole properties. During the optimization process, 55 formulations based on the HPC and PVA polymers were analyzed. However, here only 6 of them are presented. The scanning electron microscopy images have shown that in the case of the aqueous and water ethanolic solution of HPC, the bits and the fibers with bit on the string morphology were observed respectively, while smooth fibers without bit formation were obtained from the ethanolic solution of HPC and the aqueous solution of PVA. Therefore, those 2 formulations were chosen to prepare all dispersible films with aripiprazole. It was found that the type of the polymer and the presence of the API had an impact on the diameter of the nanofibers. The HPC-based fibers were thicker than fibers obtained from the PVA solution. Also, drug incorporation led to an increase in the diameter. The PVA-based films that contained aripiprazole were smooth, soft, and fluffy due to their fiber structure, while the films based on the HPC were rough, brittle, and difficult to detach from aluminum foil. Therefore, only PVA films were analyzed. The prepared or dispersible films were characterized by rapid disintegration time within 1 second, good mechanical properties, and a faster dissolution rate of aripiprazole in comparison to the raw API. I would like to emphasize that fast disintegration time is indicated as a key factor affecting patient compliance. Thank you for your attention.
Thank you. And the third presentation, also from Department of Pharmaceutical Technology and Biopharmaceutics in Kraków, in collaboration with University of Silesia. Jolanta Petera will present evaluation of factors affecting the ketoprofen release from 3DP tablets.
Hello everyone. My name is Jolanta Petera. I work in the Department of Pharmaceutical Technology and Biopharmacy at Jagiellonian University Medical College in Kraków. I would like to present you results of our work on the evaluation of factors affecting the ketoprofen release from 3D printed tablets. We prepared series of tablets with different shape, internal structures, and dosages made of polygonal alcohol-based filaments containing 20, 40, or 50% of ketoprofen and 4% of cross-covalent. We confirm an excellent properties of filament containing 40% of ketoprofen by printing 12 tablets with 100 mg of API during a single process, which you can see here on the time-lapse movie. The tablets' weight variations were within pharmacopoeial limits. Next, we compared tablets made of filaments with different drug loading. Regardless of the ketoprofen content in the filament, dissolution profiles of tablets were similar. Moreover, we analyzed the effect of internal structure. Tablets with 9.5 and 20% honeycomb infill had similar dissolution profiles because of high surface of layer adhesion in this type of infill. Round tablets containing 50 and 25 mg of ketoprofen were characterized by different dissolution profiles. Thus, we printed oblong tablets containing 50 mg of API characterized by almost the same dissolution profile as round tablets containing 25 mg of ketoprofen. In conclusion, it is possible to prepare dose-adjusted tablets with defined release profiles, but it requires structural modifications. Thank you for your attention.
The next two presentations come from the group which represents my department, Department of Pharmaceutical Technology in Gdańsk, and they are also qualified here as a SIPUS posters. Elisa Wolska will present the influence of choice of method and test condition on the results of drug release study from lipid microspheres.
Ladies and gentlemen, I would like to summarize the most important assumptions and conclusions presented on the poster entitled Influence of Choice of Method and Test Condition on the Results of Drug Release Study from Lipid Microspheres. Solid lipid microparticles are multi-compartment drug carrier which might be administered by various routes. The main advantage of SLM is the prolonged release of the drug substance. Therefore, in the case of lipid microspheres, studies on the release of the active substance are so important. Release studies play a key role not only in drug development, but also during the stability testing and quality control. Thus, the selection of an appropriate test method is crucial. Standard dissolution testing, according to Pharmacopoeia, are limited to the classical dosage forms and in most cases are not suitable for testing of modern drug carriers like solid lipid microparticles. In such a situation, it becomes necessary to select non-pharmacopoeial test method. 3 methods were used in our research: in the system without membrane, in a dialysis bag, and in side-by-side diffusion chamber. The results obtained in our release studies suggest that release of significant amount of API in a short time, which is called a burst effect, was observed only in the test method without dialysis membrane. It was shown that the tested active substances differ significantly in their ability to penetrate the diffusion membrane. Apart from the properties of the drug substance, the results of release studies are also influenced by the type of semi-permeable membrane. And finally, the faster release from the API suspension compared to the SLM dispersion confirms the effect of the prolonged release from the lipid microspheres. On the basis of the obtained results, the dialysis bag method was selected as the most favorable for the SLM examinations. Taking into account all the above factors, it should be stated that any comparative test should always be carried out using the same method and exactly the same test conditions. Thank you for your kind attention.
Katarzyna Cętkowska will present the topic of technology of oral dispersible polymer films as multi-particulate drug delivery systems with pellets, and especially the influence of different granules loading on the film properties.
Good morning, ladies and gentlemen. I would like to present results of work, technology of oral dispersible polymer films as multi-particular drug delivery system with pellets, influence of different granules loading on film properties. The research were made in the Department of Pharmaceutical Technology in Medical University of Gdańsk. Our dispersible films are single-dose drug solid dosage forms designed to be applied to the oral cavity, characterized by rapid disintegration without the need to swallow them with the water. Although the most popular approach is to prepare our dispersible films with dissolved active substance, a drug may be also incorporated in the film as a solid particle, like crystal, nanoparticles, or granules. The aim of this research was to obtain homogeneous fast disintegrating or dispersible films containing incorporated spherical granules, pellets in size of 100 to 300 micrometers. The incorporation of the pellets will probably make it possible to obtain the fast disintegrating multi-particular unit system that can be used to achieve modified release profiles or taste masking. Our dispersible film were prepared by solvent casting method. Morphology analyses were performed to assess roughness of the surface, homogeneity of the pellet distribution, and film thickness and particle incorporation. The surface and cross-section characteristic of films was visualized using scanning electron microscopy and digital microscopy. Optical microscopy image stitching mode allowed to visualize the particle distribution. The mechanical properties and disintegration time were evaluated in dependence to pellets concentration. It is possible to obtain homogeneous multiparticular or dispersible films with high pellets content up to 45% according to dry mass, characterized by an adequate uniformity of mass and particle distribution. The incorporation of solid particles into polymer film matrix shortens disintegration time significantly in dependence on the pellet size. The measurements performed with a texture analyzer are a convenient tool for assessment the influence of granule size and concentration on the mechanical properties of the films. The tear resistance and flexibility of the films were decreased by pellet presence, but still they could be packed and used by the patient. Thank you for your attention.
owe you some explanation that there was on one slide 2,000 micrometers for the pellets. They were 200 micrometers and 100 micrometers. Okay, and now we are waiting for questions. No, 2 millimeters did you use? Okay. No, we can't hear you. The online. So yes, my question is, dear Dr. Centkowska, how did you present that there are 2,000 micrometers of the pellets in the films, oral films? There was one information like this.
They were measured according to the measurements in optical microscope. We just make a stitching mode to estimate the amount of the courses per surface area. I think this is a really important one, because when we cast it with different size of the gaps, the amount of the spreaded particles inside of the surface is different, and it depends also from the size of the courses and the size of the gap. So we decide to estimate the number of them on the surface as important and crucial parameter. And it was possible in our laboratory by just analyzing the pictures, but it's also possible to use in programs to later analyze the aggregation and amount of them on the surface.
Yes, maybe more questions. Yes, Professor Breimer.
I have a question to Jolanta in Krakow. Is that possible?
Yes. Jolanta Peterav.
Yes.
Are you with us?
Now?
Yes.
So yes, good afternoon. I'm ready for your question.
I have a question for you. You showed very interesting results through the 3D printing, showing interesting different release profiles dependent on the composition of the printing material. Do you think that you can also use the printing technique to combine for example, two different drugs with different and program them for different release rates?
Yes, I'm sure it is possible. We can use it in two ways. We can use it. We can make these tablets by extrusion two filaments during the same time, but by co-extrusion of two filaments. But in this case, we will probably have a problem with controlling the dosage of each filament, because we can set, for example, that our tablet should be built in 25% made of one filament and 75% in the second one, and all structure will be built of the same mixture of these filaments. But we can also use a tool head with where we are able to put two separate filaments and, for example, put one part of tablets, print one part of tablets using the first one filament with the first active ingredient. And next, we can just continue printing with the other nozzle, printing the other filament with different API. And moreover, it is also possible to print in this case using two different temperatures. So we can change everything in this case.
Thank you very much. It sounds promising.
You're welcome.
More questions? I have a question to Ewelina Wyszczarz. Okay. Yes. You presented a very elegant product. And what problems within the industrial production you can predict?
With oral dispersible films, yes?
Because it was really presented as a very easy way and very nice way. And do you know something about the problems that you may face during industrial production of such film?
production of electrospinning by this process. Okay. Is the different apparatus, different, here in my study I used syringe and syringe pump, and in industrial production we need to use bigger apparatus, bigger syringe pump or other distribution of our solution. And also here I also use a needle around from 0.6 to 1.0 millimeters. And also I think it is for example, in production, when we prepare films in production level, probably we should use, for example, more needles, more to obtain higher bench or higher higher size of the films, and then cut them into appropriated small single dosage forms.
So maybe evaporation rate also may influence a lot.
Evaporation and the constant humidity and temperature also, it's very important during this process.
Thank you very much. More questions? Yes, Professor Breitkreuz.
I would have another question to Evelina. So as we have seen you on the screen, I think it's a good occasion now. Could you say something about the solid state of your aripiprazole within your formulation? Was it amorphous after?
Yes. However, this research is part of the bigger part of the research, and some of the results were recently published in Pharmaceutics. And yes, aripiprazole was in amorphous forms, however, only partially, because on the thermograms, it was like some recrystallization was observed. However, on the diffraction, it was fully amorphous. Therefore, we assume that some amorphization occurred during this process.
Exactly what I expected, I must say, because the drug is very poorly soluble. And yes, if you showed us this very nice video on instantaneous disintegration, I would say, do you observe later on precipitates of the drug substance?
Yes, yes. It still was around 1 second, 1.5 seconds.
Okay, but these crystals are redeveloped afterwards, right?
Yes.
They are absent in your formulation, but they reoccur in the solution, unfortunately. I would expect this. Yeah, okay.
Yes.
Thank you very much.
Thank you very much.
Thank you. Thank you very much. I can't see more questions, so thank you to all presenters at this session, and we'll go to the closing session. And first, I would like to contact with Professor Aleš Mrar from University in Ljubljana. As I mentioned in the beginning of our symposium, Professor Mrar was a godfather of this CESP symposium. So it will be really our great pleasure at the end of this symposium to listen to you, Professor Mrar. So the podium is yours.
Thank you very much, Margaret, for your nice introduction. I'm really happy to be with you. In fact, I'm with you from the very beginning, since Thursday afternoon, and I was really enjoying the presentations and how the symposium has been prepared and led. You invited me to tell a little more about the past and future of the symposium. So I have prepared a short speech to address relevant data, which led us from as early as 1995 to 2021, from Bled to Gdańsk. So, ladies and gentlemen, dear participants, I am really excited to have the opportunity to welcome you at the Central European Symposium on Pharmaceutical Technology. This is the 13th meeting, and it is really necessary to stress that the series of the events started with the aim to make a scientific upgrade of the area in the region, to establish personal contacts among scientists, to create networks, and to promote integration between academia and industry. I can freely say now, after more than 25 years, that the goals were successfully met. The best proof for this is the event in Gdańsk, where more than 150 participants were gathered to follow original aims of the symposium. I am especially pleased that we have gathered in such large number, regardless of the COVID pandemic that has been threatening us for so long and has no end in sight. It seems that we will have to live with this burden for quite some time, which, however, will not prevent us from further scientific efforts in the field of drug discovery and drug development. We are a strong and robust part of the society, and I am very pleased that our symposium is also an important part of scientific community. Moreover, I believe that this year's meeting also represents a small contribution to controlling the COVID pandemic, and maybe all together we make a step ahead. Because without the past, there is no future. I would like to provide some historical details on how CEPS came to be. In the late 1980s, the initiative for Central European scientific meetings dealing with pharmaceutical technology arose as a result of the long-standing collaboration between the departments of pharmaceutical technology of the faculties of pharmacy in Trieste, Italy, and Ljubljana, Slovenia. The first Central European Symposium on Pharmaceutical Technology was held in October 1995 in Bled, Slovenia, under the patronage of UFEPS. The subsequent events were successfully organized twice in Portorož, Slovenia, in Vienna, Austria, Ljubljana, Slovenia, Siofok, Hungary, then again in Ljubljana, Slovenia, followed by Graz, Austria, Dubrovnik, Croatia, again Portorož, Slovenia, Belgrade, Serbia, Szeged, Hungary, and with the last symposium being held these days in Gdańsk, Poland. Although the initiative was offered by the group of enthusiastic individuals from universities of Trieste and Ljubljana, the idea was very quickly adopted wider, especially in UFEPS, because it was then that UFEPS members began to be encouraged to organize regional and thematic conferences in various fields of pharmaceutical sciences. At this occasion, I would like to point out Professor Didi Breimer, the then president of UFEPS, who immediately accepted the proposal and also attended the first meeting in Bled, together with reputed professors Dominique Duchesne from France, Attila Incal from Turkey, and Professor Clara Pintier-Hodi from Hungary. Professors Alexander Flores from UK and Hans Junginger from Germany joined us 2 years later and contributed significantly to the growth of the symposium. presence of Professor Breimer at this year's symposium is a proof that the symposium continues to successfully carry out its mission. Thank you, Professor Breimer, for joining us again. I'm very delighted that you accepted our invitation and came to Gdańsk in person. Proof of a high scientific level of the symposium are also, I have to stress this very important issue, 4 thematic issues of International Journal of Pharmaceutics and 3 thematic issues of European Journal of Pharmaceutical Sciences, with published full-length papers selected as the best among oral and poster presentations. And I sincerely hope that this good practice continues. Because science is advancing so fast, 2 years ago when I retired from the university, I decided to hand over the symposium to the younger generation. And I believe that Professor Rok Drew will lead the project forward successfully. Due to continuity, I proposed that Faculty of Pharmacy, Goethe Institution, University of Ljubljana as founding institution of CESPT, set a permanent homepage of symposium with the linkage to the current organizer. I have 2 messages for you at this moment. First, CEPS today should be viewed particularly as a forum which offers open discussions on recent advances in the area, especially for younger scientists, PhD students, and postdocs. For this purpose, it is necessary to ensure the largest possible active participation of young people on the one hand, and the best experts in the field on the other, to facilitate the flow of knowledge and keep CEPS alive. Second, I am glad to announce that the position of organization of the 14th CEPS in 2023 is open, and on this basis, I am using the opportunity to invite also from my side all parties that are interested in this direction to prepare the application. Professor Drew will probably provide some details on this issue later. And finally, as far as I'm concerned, I'm still active in the area, not very much, but doing things I love and know how to manage. And now, last but not least, I would like to thank Professor Małgorzata and her group, her staff, for the exceptional organization of this symposium and for the ingenuity with which she carried out a logistically demanding meeting despite very unfriendly external circumstances. Thank you very much again, Margaret. You have done an excellent job. I wish you all the best, dear participants, ladies and gentlemen. I am Aleš Mrhar, past president of Central European Symposium on Pharmaceutical Technology. Thank you very much for your attention.
We are very grateful, first of all, that many years ago you pushed us, scientists from Central Europe, to participate in this kind of symposium, because the young people here, they don't really even imagine how it was even in the beginning of 90s. There was really a big gap between the research in Central Europe and in Western Europe, and these contacts that you created helped really many scientists to be closer to those who are more experienced in Western Europe. So we are very, very grateful for this idea, and also for the professors who helped from the Western countries to organize these meetings. Now with Rock, we will continue this closing session.
Thank you for the word. So I would firstly like to thank you, Professor Merher, for this speech, for revealing historical facts and for the prospects of the future of this symposia. So it's a great responsibility to continue your work, and hopefully we will continue with this CESP conference series. And I would also like to thank the organizer for their excellent work, and especially also for the hospitality that they have shown. I wish that every participant would be here in Gdańsk, but at least in this way, no one was left behind. So if there was a willingness to participate, one could do it. So the next symposium will take place in 2 years' time. However, the place of venue and organizer is not known yet at this point, but will be determined till the end of this year. So please come forward to me if you have a preference and an ambition to organize and host the next symposium. I'm sure that we will find an organizer. So finally, I would also like to touch the dilemma whether future symposia should cover broad topics like this one at this point. So the benefit of having broad range of key topics is that almost no one is excluded from potential participation. However, we have to have support from the departments across Europe to achieve critical number of contributions for a specific theme. I also believe that that listening to research achievements from broader range of topics can give us that crazy idea, or at least give the necessary perspective for a future research project. As pharmaceutics is in its core an applied science, we always have to have a good answer why we are doing a particular research, investing a lot of time and effort. So I still believe that we should have such rather broad topics involved in this kind of symposia. So with this, I guess we are now concluding this symposia. I wish you all a pleasant trip back to your homes, and hopefully we will meet all together in live version in the next 2 years' time. So thank you.
Thank you, Rob. Few more words from myself. Dr. Schliau today said how it's wonderful to meet old friends and then to make new friendships, and I really share this opinion, because we really, I think we get this feeling also here, and I have a feeling that also you online, you were much closer than maybe you thought, because we really appreciated the presence of everybody online. And I appreciate very much all this who decided to present or lectures or these even short poster presentations. I am very grateful for everybody who presented or sent these recorded lectures. I know that it's also difficult to find time even to record these lectures. It's an effort. So thank you. And I would like very much to express my thanks to to the professors who were really supporting the idea and our preparations, because not only Rok Mrar and Ales Mrar and Rok Drow, but also I've got support with the letters from you, like Stane or Andreas. It was really very, very important for us to go on with this support. Of course, I would like to thank the group in our department who helped to make this job, but of course, my deepest thanks go first of all to Bartek Maciejewski, the secretary of our symposium. Almost all this work was done by him with support of his wife, Bianca, and also with the support of Basia Mikolaszek, Eliza Wolska, and also others. Rok mentioned that it was probably difficult in this unfriendly atmosphere. I would say that I don't feel so much that it was unfriendly, just opposite. Even during preparations, we felt quite comfortable, I would say. I was suspicious why it's going on, but it was very much thanks to really a wonderful collaboration with Adept Company and Michał Kaszyński and his wife Ewa Kaszyńska. Really great, great support, and maybe you will come and show yourself to the camera, because the online, so please stand here, because our really the people who are online should know your faces, that it's really thanks to you. Thank you very much. I would certainly recommend that. It was ideal collaboration. And thank you very much for the sponsors. They, since the very beginning, they are really willing to help us. This is Polpharma. Always very supportive, and as you know, the biggest generic Polish company, and also so close to Gdańsk, and we have really wonderful also contacts all the time, and the company Aptar and Natoli and Kiens and ITS and Servier. So thank you all the sponsors. Yes, I wish closing. I wish everybody all success in the research. So till the next our meeting, CESP meeting, we will have more and more results, and another meeting will be even more successful. I believe that the presentations during this symposium were really on a high level, and this satisfies me really a lot. So I'm personally, because of that, very happy. I wish you a safe, of course, trip home to those who are here, and for you online, just a nice weekend. And also, I should express my hopes that we'll meet in the future, in the nearest even future, and not only at this symposium, but I always admire Professor Jörg Breitkreuz and APV, how wonderful, huge congresses you are organizing for us. So we are looking forward also to meet there. So thank you once again. I hope I didn't forget about something, but usually it is a case, so I'm sorry. Thank you very much. You will see us still on the turn, please, the camera, so people online will see that we are still here and we are ready for lunch. Thank you. Goodbye.